So picture this. You are standing in the ER and a patient is rushed in. They've got severe tremors, slurred speech, and they are, rapidly slipping into a coma.
Right, and your first instinct in that scenario is probably trauma, right? Like a stroke or maybe a severe drug overdose.
Exactly. But the actual culprit is just a microscopic, totally everyday byproduct of, like, eating a steak.
Yeah, a tiny molecule of ammonia.
Right, an ammonia molecule that the liver basically failed to clear. So it crosses the blood-brain barrier and completely shuts down the brain's cellular power grid.
It's wild. That is the raw, high-stakes reality of human biochemistry.
Yeah.
I mean, it isn't just some list of invisible pathways you memorize for a
Well,
test.
no, it's the fundamental machinery of life and death.
It really is the ultimate puzzle, because every single breath you take, every muscle twitch, every immune response, it's all governed by these chemical reactions that are happening at just a staggering rate.
Yeah.
And when even one of those reactions misfires.
Like an enzyme is missing or something.
Exactly. One enzyme goes missing or one molecule builds up, and the entire system just starts flashing warning lights.
Okay, let's unpack this, because our mission today is a really comprehensive, scientifically rigorous deep dive into this incredible masterclass, Oraby's illustrative reviews of biochemistry.
A fantastic resource.
It really is.
Okay.
And we have tailored this deep dive specifically for you, right? So whether you're the medical student prepping for boards, a young professional in the health sciences, or just, a fiercely curious learner who wants to truly master the mechanics of the human body.
Because we are going to track the entire biological supply chain today.
Yeah, from the moment dietary proteins get shattered into building blocks to the pressurized fluid networks that distribute them.
Right, and the strict chemical environments that keep them from falling apart, all the way down to the genetic master code that dictates their construction.
And we aren't just going to list off what these components are either. We're going to look at the why and the how.
Well, yeah, because understanding the mechanism is really the only way to recognize the pathology.
Exactly.
When you know how the machine operates under, like, perfect conditions, a disease stops being this mysterious list of symptoms. It starts looking like a very specific mechanical failure.
A failure that you can actually fix.
Exactly.
So let's start right at the intake valve, right? Digestion. You eat a piece of chicken. That protein is this massive, complex, folded chain of amino acids.
Yeah, it's huge.
And it's completely useless to your bloodstream in that state. It has to be dismantled. So it hits the stomach, which is just bathed in hydrochloric acid, dropping the pH drastically.
Right.
And that acid bath activates a proenzyme called pepsinogen, turning it into pepsin. And pepsin then acts like a pair of biochemical scissors.
But not just any scissors, though. Pepsin is an endopeptidase.
Right, so it cuts from the inside.
Exactly. It doesn't just nibble away at the ends of the protein chain. It attacks the structural integrity by cleaving the bonds right in the middle.
Just shattering it.
Yes, specifically targeting the bonds formed by aromatic amino acids. It breaks those large polypeptide chains into much smaller, manageable fragments.
Which is so cool. But I want to look at how this changes early in human development, because an infant's stomach isn't this highly acidic vat yet, right?
No, not at all.
And their primary protein source is milk casein, which is liquid. So if they just relied on pepsin, that milk would basically wash straight through the digestive tract before any nutrients could even be extracted.
Right, it would move too fast.
So they use an entirely different enzyme called renin. And renin literally coagulates the milk.
Mechanistically, yeah, that's exactly what it does. Renin interacts with the milk casein in the presence of calcium, and it alters the structure.
Turning it into a clot.
Right,
Yeah.
an insoluble clot known as calcium caseinate.
So an infant's stomach essentially turns milk into a temporary cheese clot just to slow down digestion,
Exactly.
which gives the enzymes the time they need to work, and it keeps the baby feeling full.
It's a brilliant adaptation.
It really is. So once these proteins, whether from the adult's chicken or the baby's milk, once they're broken down further in the intestine by pancreatic enzymes like trypsin and chymotrypsin,
Right.
they get absorbed as single amino acids. And they enter the body's amino acid pool, which sits at about 100 grams of free amino acids at any given time.
Oh,
But we don't have a storage tank for proteins like we do for fat, do we?
we don't. That's a crucial point. If an amino acid isn't immediately recruited to build new muscle tissue or synthesize an enzyme or something, it faces the biological chopping block.
It gets stripped down.
Yeah. The body removes the nitrogen-containing amino group through transamination, which is passing the nitrogen to another molecule, or deamination, which just strips it off completely.
And that stripping process creates our first massive biological exhaust problem because deamination produces free ammonia.
Yep.
And ammonia is essentially physiological poison.
It is. And what's fascinating here is the sheer elegance of the liver's triage system.
Oh, the urea cycle.
Exactly. The liver runs the urea cycle specifically to neutralize this threat. It pulls ammonia and carbon dioxide into the mitochondria, and it essentially forces them together to create a compound called carbamoyl phosphate.
Right.
And through a highly coordinated cycle of chemical intermediates, it converts that volatile ammonia into urea, which is, water-soluble, it's stable, and it can be safely filtered out by the kidneys.
But let's go back to our ER patient from the very beginning, the one with severe liver disease.
Ah, right.
So in their case, the urea cycle is offline, the ammonia levels surge, and because it's a really small uncharged molecule, it slips right past the blood-brain barrier, and the brain just panics.
It completely panics, and it initiates this desperate and ultimately fatal defense mechanism.
To neutralize the ammonia.
Right. To neutralize it, the brain grabs a molecule called alpha-ketoglutarate and fuses it with the ammonia to form the amino acid glutamate.
Which sounds like a smart move at first.
It does, until you realize what alpha-ketoglutarate actually is. It is a critical, completely irreplaceable intermediate in the citric acid cycle.
The Krebs cycle.
The Krebs cycle, yeah.
And the Krebs cycle is the cellular engine, right?
Okay.
It generates ATP, the energy currency of the entire cell.
Exactly. So by cannibalizing alpha-ketoglutarate to fight the ammonia, the brain completely stalls its own citric acid cycle.
Wow.
ATP production just plummets. The neurons literally run out of electrical power. And that energetic collapse is what drives the tremors, the slurred speech, and ultimately the hyperammonemic coma.
It is just a phenomenal example of a short-term fix causing a catastrophic systemic failure.
It really is.
Now, ammonia isn't the only toxic waste the liver has to juggle. We have to talk about heme.
Right.
Red blood cells are essentially the cargo ships for oxygen, and they use the iron-containing heme ring in hemoglobin. But those cells take a serious beating. They only survive for about 120 days before they're just too battered to function.
Right, they get worn out.
So the body has to tear them down and recycle the parts.
And the reticuloendothelial system handles that demolition. The iron gets salvaged, the globin proteins are broken back down into that amino acid pool we talked about.
Yeah.
But the heme ring itself is cracked open, and it turns into this green pigment called biliverdin, which is then really quickly reduced to a golden yellow compound called bilirubin.
And here's the mechanical flaw, right? This raw, unconjugated bilirubin is lipid soluble. It's basically molecular grease.
That's a great way to put it.
And the kidneys cannot filter grease into the urine. You can't just flush it out. So the liver has to act as a chemical modification plant.
Right, it has to process it.
Yeah, it takes this unconjugated bilirubin and attaches a sugar acid to it. It conjugates it. And that single chemical modification waterproofs the bilirubin so the body can safely secrete it into the bile, down into the intestines, and out of the body.
Yep.
But let's look at the diagnostic side of this. Say a patient walks in and their skin and the whites of their eyes are distinctively yellow, jaundice. We know the bilirubin is backing up into their tissues, but how do you know if the liver itself is failing or if the plumbing is just blocked somewhere?
Well, clinically, we use the Vandenberg reaction.
Okay.
This test distinguishes between those two types of bilirubin we just mentioned. If the blood shows incredibly high levels of direct or conjugated bilirubin, the diagnosis is pretty clear.
The liver did its job.
Exactly. The liver successfully waterproofed the waste. It did what it was supposed to do. But the waste couldn't actually exit the body, likely because of some downstream blockage, like a gallstone stuck in the bile duct.
So the plumbing literally backed up.
Right. But if the test shows high levels of indirect, unconjugated bilirubin, then you know the problem is upstream.
Ah, okay.
The liver's processing plant is either failing due to some disease, or the red blood cells are being destroyed so rapidly in a state of severe hemolysis that the liver is simply overwhelmed by the raw volume of material.
Incredible. So just by tracking the exact chemical state of the exhaust, you can pinpoint exactly which part of the machine is broken.
Precisely.
Now, this brings up a logistical issue. We have all these raw materials, right? Amino acids, newly synthesized enzymes, conjugated waste. If they just sit in the liver, they're useless.
Yeah.
The body requires a highly specialized, pressurized distribution network.
Yes, the liquid landscapes.
Yeah.
Body fluids are far more than just, water sloshing around.
Yeah.
They are highly engineered mediums tailored for really specific environments.
Yeah.
We have the sheer transport volume of blood, the waste removal channels of urine, and specialized fluids like cerebrospinal fluid that buffers the
Or
brain.
just novial fluid.
Oh, synovial fluid is fascinating. It's in your joints, and it's packed with hyaluronic acid, making it incredibly viscous.
Yeah.
It acts as a high-grade biological lubricant, so your bones don't just grind against each other.
And because these fluids circulate everywhere, they act as diagnostic windows into the health of solid organs. Like if an organ takes damage, its internal cellular machinery literally spills out into the fluid highways.
Right.
So say a patient comes in clutching their chest, you draw blood and look for an enzyme called CKMB.
Because CKMB normally belongs inside the muscle cells of the heart.
Exactly.
So if you find it floating freely in the blood plasma, it means those heart cells have ruptured and died. It's a definitive biomarker for a recent myocardial infarction, a heart attack.
Right.
And similarly, if enzymes like AST and ALT are elevated in the blood, you are generally looking at cellular necrosis in the liver.
We look for anomalies in urine too, right? A healthy kidney shouldn't let large proteins or sugars slip into the urine.
Never.
If you detect ketone bodies, ketonuria, it means the body has lost the ability to utilize glucose and is just aggressively burning through fat reserves,
Right.
which is a classic hallmark of uncontrolled diabetes.
Very true.
Though I guess it's worth noting some things are supposed to be there, like Tam-Horsfall mucoproteins are naturally secreted by the renal tubules, so you can't just look for what's wrong, you really have to know what normal looks like.
Absolutely. And normal in these fluids includes a massive standing army. Floating within the blood and mucosal secretions are our ultimate defensive proteins.
The immunoglobulins.
Yes, our antibodies. Structurally, they're just brilliant. They're shaped like a Y, built from 2 heavy chains and 2 light chains. And we categorize them into 5 main classes: IgG, IgA, IgM, IgE, and IgD.
Okay, here's where it gets really interesting for me. The text dives into neonatal immunity.
Oh, this is a great section.
Because a newborn baby has an immune system that is essentially a blank slate. It has zero memory of the pathogens waiting for it outside the womb. So how does it even survive the first few months?
Right.
It survives entirely on maternal hardware.
It does. During pregnancy, the fetus is shielded because IgG is uniquely adapted to cross the placental barrier.
Wow.
It is the only immunoglobulin that can do this, granting the fetus the mother's systemic immune memory. But after birth, the whole strategy shifts.
Right, because of the milk.
Yes. The mother produces colostrum, the early breast milk, which the text sometimes refers to historically as witch's milk.
And colostrum is absolutely loaded with IgO. So while IgG was protecting the internal bloodstream, the baby swallows this IgA and it physically coats the lining of the infant's entire gastrointestinal tract.
It's amazing.
It acts like this protective biological paint, neutralizing pathogens before they can even cross into the baby's system.
It's a 2-stage deployment system that basically ensures survival. And, just to complete the major players here, IgE is the antibody we all curse during allergy season.
Right.
Because it binds to seemingly harmless allergens and triggers mast cells to just dump histamine, causing all that inflammation.
No. Antibodies, enzymes, transport proteins, they are incredibly fragile machines. They rely entirely on their precise 3D folded shapes to function.
Completely.
So if the environment they float in becomes like too acidic or too basic or lacks the right minerals, they denature, they unfold and just become useless garbage. So the body has to act as a strict chemical stabilizer.
And this requires rigid mineral metabolism. We have macro minerals like calcium, phosphorus, sodium, and potassium, and they govern fluid balance and structural integrity.
Right.
And then we have microminerals, or trace elements, which act as vital ignition keys for specific proteins, like we already mentioned iron for our heme rings.
Yeah.
We need iodine to synthesize thyroid hormones, and copper for mitochondrial energy transfer.
Calcium is actually a great example of this tight regulation. Because it isn't just structural concrete for bones. Free calcium in the blood is the trigger for muscle contraction and blood clotting.
Exactly.
If your blood calcium drops, your heart muscle literally can't fire properly. So the body uses this 3-part system to micromanage it.
Right.
Vitamin D acts on the gut to ramp up calcium absorption from your diet. But if that's not enough, parathyroid hormone, or PTH, acts like a biological drill.
I love that analogy.
Yeah, it pulls calcium directly out of the bone bank to keep blood levels stable, and then calcitonin acts as the emergency brake, actively lowering blood calcium if it spikes too high.
It's a beautiful system. But balancing minerals is actually simple compared to the most critical stabilizing act in human biochemistry, which is acid-base balance.
Oh, man. Yeah.
The pH of normal arterial blood must stay strictly between 7.35 and 7.45. A drop to 7.0 or a rise to 7.8 will shut down the enzymatic functions of the body entirely. It is just fatal.
And this raises an important question, though. The human body is a massive acid-generating factory.
Oh, huge.
Every time a muscle flexes, every time a cell burns glucose, it spits out acidic metabolic waste. So how do we prevent our own exhaust from constantly dragging our blood pH down to lethal levels?
Well, we rely on chemical shock absorbers, buffers.
Okay.
And the most crucial one in our extracellular fluid is the bicarbonate and carbonic acid buffer system. And it operates on a very strict mathematical mandate.
The 20-to-1 ratio.
Exactly. To maintain a perfect pH of 7.4, the ratio of bicarbonate, which acts as the neutralizing base, to carbonic acid must be maintained at exactly 20-to-1.
20 parts base to 1 part acid, and the body assigns 2 massive organ systems just to manage this one ratio, right?
Yeah. The lungs are the rapid response unit, controlling the carbonic acid side of the equation just by exhaling carbon dioxide.
Right.
While the kidneys are the heavy-duty managers handling the bicarbonate side, the kidneys actively reabsorb bicarbonate back into the blood so we don't lose our base, and they forcibly excrete excess hydrogen ions into the urine.
Oh, and they use ammonia for that, right?
They do. They take that toxic ammonia we discussed earlier, bind a hydrogen ion to it, and excrete it safely as ammonium.
So let's see this in action clinically. Take a patient with severe, uncontrolled diabetes.
Okay.
Because they can't utilize glucose, their liver starts churning out massive amounts of ketone bodies to use as alternative fuel. But ketone bodies are highly acidic, so this floods the bloodstream with acid, rapidly burning through the bicarbonate reserves. The 20-to-1 ratio just plummets. This is metabolic acidosis. So if you walk into that patient's room, what is the very first physical sign you are going to see?
You are going to see a really desperate attempt at respiratory compensation. The patient will be taking massive, rapid, deep breaths. It's a pattern known as Kussmaul respirations.
Right.
And they aren't doing this because they lack oxygen.
Right. It's about the acid.
Exactly.
Yeah.
Because the metabolic side, the bicarbonate, has crashed. The lungs are trying to fix the ratio from the other side.
By blowing off CO2.
Yes. By hyperventilating, they're blowing off huge amounts of CO2, which drastically lowers the carbonic acid levels. The goal is to force that fractional ratio back toward 20-to-1 just to buy the body time.
It's a dynamic, high-stakes physiological seesaw.
It really is.
Okay, so we have covered the protein fuel, the fluid highways, the mineral stabilizers, and the pH buffers.
Yep.
But none of these intricate protein machines build themselves, right? An antibody doesn't just know how to be a Y shape.
No.
A red blood cell doesn't just guess how to assemble hemoglobin. The exact specifications for every single piece of biological hardware must be coded, stored, and read.
And now we were talking about the molecular blueprint, nucleotides and nucleic acids.
Yeah.
A single nucleotide is built from 3 distinct chemical components, right? A nitrogenous base, a pentose sugar, and a phosphate group. You string billions of these together, and you have DNA and RNA.
Right.
But much like proteins, nucleotides have their own metabolic life cycles and their own toxic waste products.
Exactly. When the purine bases, specifically adenine and guanine, are broken down by the body, they don't form urea, they form uric acid.
And uric acid has incredibly poor solubility.
Right.
So if a patient's metabolism overproduces purines, or more commonly, if their kidneys fail to filter out enough uric acid, the concentration in the blood rises until it reaches a physical breaking point.
It precipitates.
It precipitates right out of solution. It forms these sharp microscopic sodium urate crystals. And these microscopic needles deposit into the joints and soft tissues, forming dense nodules known as tophi.
And the immune system sees these needle-like crystals just tearing up the joint lining and launches a massive viral inflammatory attack.
Yeah.
Clinically, we know this as gout.
Gout.
It classically presents in the dead of night as a red, swollen, agonizingly painful big toe. It's really just a physical manifestation of a microscopic waste clearance failure.
It's awful. But, the true majesty of nucleotides isn't in their breakdown, it's in their sequence.
Right.
The flow of biological information is dictated by the central dogma of molecular biology. DNA transcribes to RNA, which translates into protein.
I always think of it like an industrial 3D printing operation, right? Your DNA is the master CAD file, the irreplaceable source code locked safely inside the nucleus.
That's a great way to think of it.
Yeah, and when a cell needs to manufacture a protein, it cannot risk damaging the master file. So an enzyme unzips the DNA and transcribes a temporary working copy of that specific section.
The mRNA.
Exactly. That copy is messenger RNA. The mRNA travels out of the nucleus to the ribosomes, which basically act as the physical 3D printers.
Right.
The ribosome reads the mRNA code, translating every three-letter sequence, every codon, into a specific amino acid, linking them chain by chain until a fully functional protein pops out.
And if we connect this to the bigger picture, right, you suddenly realize how terrifyingly fragile the system is.
Oh, yeah.
Like, what happens if there is a typo in the master code? Look at sickle cell anemia. The DNA blueprint for the hemoglobin molecule contains a single-point mutation.
Just one letter.
Out of billions of letters, just one is swapped. But that changes the mRNA codon, which forces the ribosome to load the wrong amino acid.
Wow.
At exactly position 6 of the hemoglobin beta chain, the normal amino acid, glutamate, is replaced by valine.
And here is where the basic chemistry dictates the pathology.
Yep.
Glutamate is polar, right? It interacts perfectly with the water in the blood. But valine is nonpolar. It is extremely hydrophobic. It literally hates water.
Exactly. And because of that single hydrophobic valine swapping in, whenever oxygen levels drop, the hemoglobin molecules try to hide from the water.
Oh, so they stick together.
Right. They stick to each other. They polymerize into long, rigid, microscopic fibers inside the red blood cell. And these fibers physically distort the cell, warping it from a smooth disc into a rigid crescent sickle shape.
And those rigid sickled cells can't bend to fit through the tiny capillaries. So they create a molecular traffic jam, cutting off blood flow and causing agonizing pain and ischemic tissue damage.
Just from one wrong letter.
One wrong letter out of billions, and the entire systemic transport network collapses. It's terrifying. But, we aren't just passive observers of this code anymore.
No, we're not.
The text closes by exploring recombinant DNA technology. We now have the actual tools to read, amplify, and edit the source code.
And the clinical leaps are just staggering. We use polymerase chain reaction, or PCR, to take a minuscule, barely detectable fragment of DNA and amplify it millions of times so we can actually study it.
Right.
And we have harness restriction endonucleases, which are bacterial enzymes that function as programmable chemical scissors.
They hunt for specific palindrome sequences in the DNA, right? Sections that read the same forward on one strand as they do backward on the opposite strand.
Exactly.
And they cut the helix right at that exact location.
And by utilizing these molecular scissors, we can cut a human gene, for instance, the gene that codes for insulin, and splice it directly into the circular DNA plasmid of a bacteria.
That's incredible.
We basically hijack the bacterial ribosome, turning it into a microscopic biological factory that pumps out pure human insulin for diabetic patients. We have literally learned to rewrite the machinery.
So what does this all mean? We started at the macro level, watching stomach acid and pepsin dismantle a meal. We traced the exhaust through the urea cycle to prevent our brain's power grid from failing.
Yep.
We watched conjugated bilirubin navigate the bile ducts and colostrum-derived IgA antibodies paint the newborn gut. We balanced the absolute chemical knife edge of a 20-to-1 buffer system, and we drilled all the way down into the nucleus, finding that a single molecular typo can physically warp a cell.
We covered a lot of ground.
We did. And it all means that whether you are diagnosing jaundice, pushing IV bicarbonate to stop Kussmaul respirations, or genetically engineering insulin, biochemistry isn't just abstract. It is the fundamental language of patient care.
It is the definitive why behind every single clinical what. And, to ensure you've truly mastered how these systems interconnect, let's put it to the test with a quick clinical review question for you.
Oh, perfect.
Listen closely. A patient is rushed into the emergency room experiencing a severe acute asthma attack. Because their airway is critically constricted, they cannot exhale properly and are retaining massive amounts of carbon dioxide in their lungs and bloodstream.
Okay.
Based on everything we've unpacked today regarding chemical stabilizers, what specific acid-base imbalance is this patient experiencing, and what exact ratio in the blood buffer system is primarily disrupted?
I'll give you a second to think about the mechanics of retaining CO2.
So if you trace the mechanism in deduced respiratory acidosis, you have mastered the concept.
Yeah.
The retained carbon dioxide acts directly as an acid in the blood, surging the carbonic acid component and destroying that vital 20-to-1 bicarbonate to carbonic acid ratio.
It all comes back to the math. But, before we sign off, the final chapter of Oraby's text opens a door that pushes past pure molecular mechanics.
It does. With the completion of the Human Genome Project and the rapid advancement of recombinant DNA technology, we have achieved unprecedented power.
Yeah.
I mean, we can pinpoint the exact nucleotide swap that causes sickle cell. We can engineer transgenic organisms to manufacture our medicines. But this raw capability demands we ask a much harder question.
Which is,
As we perfect the ability to isolate and manipulate the human code, where exactly is the line between utilizing this knowledge to cure a devastating disease and commercially patenting the very genetic sequences that make us human?
wow. We finally have the master blueprint in our hands. Now we have to figure out who owns it. Keep unpacking that one.